This video shows the difference in quality of peaks between the forward and the reverse strands during a sequencing. So it means if I make my consensus sequence by aligning the two sequences, the forward and the reverse complement, I should be getting similar letters in my arrangement or alignment. So as we can see, there is A here, there is A, there is A, there is A, but here there is C, there is A, meaning there is a mismatch here. So we need to make a correction during the formation of a consensus sequence. So if we are to correct this, where should we go, or which sequence should we rely on for the best correction? So if we visit the same region in the reverse and the forward, in the forward we can see they are very good peaks. They are very sharp and separate. If we come to the reverse, we can see the peaks. They are not as sharp and separated as compared to the forward. So I would go for the forward strand. See what I'm saying? This is the forward, this is the reverse in the same region here. So meaning if I'm to correct this, I would correct according to the reverse strand, as we can see. As we move further to along the sequence, after aligning, we shall see that around region 520, after correcting the reverse and the forward, making the consensus, you can see that around this region there is a mismatch between T and G. So T and G, which strand should we go to, reverse or forward? If we come around the same region and check the quality of the sequences, the forward and reverse, can witness that the reverse has better sequence compared to the forward. So I would correct this region, depending on my reverse strand. If you go further on the sequence still, you can see that the bands here, they are not as sharp as here. So I would correct also this region here, as highlighted here, the region around 610, depending on my reverse strand in preference to the forward strand, because the reverse strand has better sharp and separate peaks compared to the forward strand. So I would make my correction depending on my reverse in this region. So in a nutshell, as we can see, that as you move from 330 or 300 region, 500 region, 500, 600 region, the forward strand the forward strand is better or has better sequences in the first zones of 300, 500, the quality reduces, and so on. So I would recommend that during formation of a consensus sequence, especially for 16S, we can see that the enzyme becomes weaker as it tends to the end. So it tends to produce less quality sequences compared to the beginning. So the main purpose of this video was to show you how to align two sequences and make corrections when you have bad sequences with a lot of mismatches. Thank you very much. Subscribe, comment, like, and share. Thank you.